mandatory pause. Okay, well, it's really an honor to get to talk to this august group today. I've obviously been following the work of Dr. Baron Cohen and the whole center for many, many years. So I'm really, really delighted to share my data with you and hear your thoughts about it. So immune origins of sex differences in the brain. Where am I coming from? Oh, come on. So I hope you all recognize Kermit the Frog from Sesame Street, who always said it wasn't easy being green. And I say it's not easy being male. And that sounds like such an apocryphal thing to say. Why would you say it's not easy being male? Well, that's because males are more likely to be born prematurely. They're more likely to suffer an injury at birth. And if they do suffer an injury at birth, they're going to have a worse outcome than their sister who might have the exact same injury. But more even broadly, and I think more impactfully, they're more likely to be diagnosed with a neuropsychiatric or neurological disorder that has origins in development, which I know is not news to you all. This was sort of my pizza pie diagram of a lot of the gender bias and various disorders. Start on the right-hand side with those that are male biased, which I note that they are all childhood in onset. So attention deficit hyperactivity disorders, autism, obviously 4 to 5 times more frequent in boys. Dyslexia and stuttering are actually neurological disorders that are about 2 times more frequent in boys, Tourette's, and then early onset schizophrenia. The left side is the female bias, all of which are by and large do not occur until puberty or afterwards. So we have late onset schizophrenia in like the 40s and 50s, anorexia, which is very dimorphic, about 13 times more frequent in women as men, bulimia, multiple sclerosis is a neuroimmune disorder, PTSD, anxiety, depression are all about 1.5 to 2 times more frequent in females. And this difference between the bias in childhood onset in males and adult onset in females, to me, really compels us to have to get to the biological origins of the sex differences in the brain. So how are sex differences determined? There's a variety of ways to get there. One is your genetic chromosome, sex chromosome complement, X versus Y. Your experiences from the moment that you're born, you're going to be talked to differently, handled differently, etc. But to my mind, one of the most important things is your hormones, which again is no news to this group. And as you know, testosterone is both a active hormone itself, but also a precursor via aromatization to 3 different types of estrogens, which are all very potent as well in their impact on the developing brain. So when it comes to humans, how do we ever begin to separate the biological influence of sex from the really powerful influence of culture and environment, which, as I said, just begins from the minute that you're born? And I think it's going to impart itself upon your brain as it develops. And the way we do that is we have to turn to our animal models. And these are some of the ones that have been the most popular over the years. My animal model is actually this guy here, the laboratory rat, and not the mouse. And there's a really good reason I stick with the rat, which we'll come to in a little bit. So how does this hormonal programming occur? Again, I don't want to bring coals to Newcastle because I know you're all familiar with this, but it's really worth pointing out the importance of these sensitive periods of development, which in rodents begin about embryonic day 18, when the male fetal testis begins to make copious amounts of testosterone. The same thing is happening in humans in the 2nd trimester. Actually, I just was reminded from reading Madeline's paper this morning, it actually begins as early as 8 weeks gestation, that we get very high testosterone in males. The sensitive period refers to that time during which a female will remain sensitive to exogenous hormone treatment. And in our rodents, we're really lucky that that sensitive period extends into the first week of life, so that we can take a female and we can inject her directly with testosterone, and we can essentially convert her into a male, just her brain, right? Not her body, because her body's already sexually differentiated. And so this is the tool that we use to try to get at the mechanistic basis of sex differentiation of the brain. So it used to be in the old days when I started, the only differences in the brain that anybody thought about that were relevant to males and females were reproductive endpoints, and all of the emphasis was on reproduction. But we now know that any endpoint in the brain during development that you can think of is in some way at some time modulated by sex. And this is sort of my summary diagram of everything in neuroscience. So we start with formation of the neural tube, which can actually be impacted by an X versus XY genotype, neurogenesis, gliogenesis, migration, dendritic growth, myelination, phenotypic differentiation, synaptogenesis, synaptic pruning, apoptosis. Boom, that's it. That's all of neuroscience, right? I defy you to name one other process that isn't named here. And I can give you a story of each one of these being modified in some way. I am going to focus today just briefly on some old studies we've done on synaptogenesis, and then some newer studies on neurogenesis, gliogenesis, and apoptosis. So how are these modifications achieved? That is the more, to me, the more relevant question, because that's how we're going to understand when they go wrong, is if we know how they are established normally. So it's multifactorial. There are things like promoting excitation via glutamate and depolarizing GABA, enhancing growth and survival by upregulating growth factors. There's steroid-induced epigenetic modifications. There are X and Y chromosome genes that seem to be important. And then there's harnessing the immune system. What? Harnessing the immune system? When did harnessing the immune system become important to sex differentiation in the brain? And I will say, I never in a million years would have ever thought that I would be thinking about immune cells, much less going around starting to call myself a neuroimmunologist. But it all came about naturally by just doing blue sky discovery research. We were interrogating one system in detail, which is that we were focusing on this brain region called the preoptic area. It's named preoptic not because it has anything to do with vision, just because it's pre to the optic chiasm. And it is sort of ground central for male sex behavior. If you lesion this brain region, males lose all interest in sex. If you stimulate this brain region, males lose all interest in anything but sex. And so we wanted to find out about, was there a morphological, a neuroanatomical substrate in this brain region that was important to that dichotomy. And what happened along the way was, still to this day surprises me, is that a young MD PhD student in the lab started thinking about prostaglandins. I was like, what? What do you mean prostaglandins? But long story short, he said, I think that they might be important to the development of the preoptic area. And this is a small lipid membrane-derived signaling molecule, very short acting, that is normally associated with making you sick, right? This is what your paracetamol, your Tylenol, your Aleve, your Advil, all of your painkillers are acting on blocking this from being synthesized. It's actually the chemical in your body that gives you a fever. And what Stuart found was that Stuart Amato was the student, rather than injecting testosterone into a female, if he just injected PGE2 directly into her brain twice on the day she was born, and one day later, he could completely reverse what should in the adulthood be female typical behavior into male typical behavior. And you just couldn't even tell these females from males. They, if we could think that they knew, they thought they were males. Now we had to give them testosterone, but a normal male has to have testosterone in order to show male mating behavior. And we published this back in 2004. And it was the first time that the masculinization of the brain had been, the steroid had been substituted for a single signaling molecule. Then a new graduate student came into the lab, and he discovered that a single injection of PGE2 on the day of birth was sufficient to masculinize the synaptic profile, the spine density, and the behavior in adulthood. So we have like a 60-day difference between we exposed to PGE2 and in adulthood, they act just like males. And that was just crazy. I mean, how could that be? That just seemed like Mother Nature playing with fire. And so we said there must be something else going on. There must be a positive feed forward mechanism. We must be lighting a fuse, basically. We must be turning on a reaction that is going to endure for some period of time in order to have this profound effect on the brain. And so we asked, is there another cell type that might be making prostaglandins? The enzymes that make prostaglandins are very ubiquitous and found everywhere. And this was right around the time that there was a reemergence, so a renaissance of the discovery of the brain's own immune system, which is now we know is the microglia. Microglia are the most unfortunate named cells in the brain. And that was because they were named by Hugo Ortega. I think it was, I might say that wrong, but he was a student of Ramon y Cajal. And he noticed on Golgi stains that these cells looked like astrocytes, but they were smaller. So he called them microglia, but they are anything but. First of all, they're not micro, they're about 10% of your brain's content. And secondly, they are not glia at all. They are the primary immune competent cells of the brain, and they are modified macrophages, which originate in the embryonic yolk sac, migrate into the brain, and stay there as a permanent resident. We were interested in them because they both respond to and produce prostaglandins, because they're always been considered to be there in response waiting for an injury. That was their role. And they also have this great morphological variety that an individual cell will change its shape, and that's actually kind of a hint into its mood. So when they have this sort of amoeboid blebby-like shape, they are, quote, activated. Oh, I misspelled activated. I got to remember to correct that typo. Versus if they are in this filigreed, fine, ramifying appearance, they are surveying, meaning they're checking on everything that's going around them and everything in between. But this is more than just what their mood is. It's actually what kind of chemicals they're producing. And so when they're in that activated state, they are producing PGE2 as well as cytokines, TNF, alpha, IL-10, etc. So one of the important features of microglia is how early they get into the brain. In our rodent models, they get in about embryonic day 9.5, which means that they're there sort of ready and waiting before all of these other critical developmental processes go on that we all care so much about, like neurogenesis, etc. It also means that they are there well before the sensitive period for sexual differentiation. And they come into the brain through the yolk set, through the bloodstream, and then they colonize the brain along the vasculature as the vasculature matures. So we had asked the question of whether or not those microglia showed a different activational state in the male preoptic area versus the female preoptic area in the heart of the sensitive period. And this is a staining for IBA1, which is a microglia specific marker. And you can see over here on the right, the female, her microglia are all in that surveying morphology. They are not activated. They're actually just being busybodies checking on everybody around them. Whereas you look at the male, they're all very blebby. They're more to, the amoeboid is, they're not moving. They just have a blebby amoeboid-like shape, and they're much, much more activated. So we in fact showed that the microglia activation state is critical to this masculinization process. So that we also published that a while ago. But you know, microglia, if you're an immunologist, you don't even mention microglia. They don't even warrant your attention at all. And all of us who are studying microglia now consider ourselves neuroimmunologists. But this is like a typical immunology chart, right? They don't care about microglia. They care about B cells and T cells and platelets and everything like that. But they also care about this one little cell. He's always down there in the corner, small but mighty, which is the mast cell. So what are mast cells? And for reasons I won't go into, we had a reason that we wanted to start looking at mast cells. They're a completely different immune cell that originates in the bone marrow from a myeloid lineage instead of the macrophage lineage. They're all over your body. They like surfaces, so they are in your eyes, your skin, your mouth, your nose, and they are the central allergic responders. They will give you itchy eyes and runny nose and rashes and everything. So they like to be on surfaces. They're packed full of these dense granules, which upon stimulation, they will release, and they degranulate and they send out these signaling molecules. And often in the context of injury, to call in the microglia after there's been, a bunch of, blood splattered all over the brain, they're gonna come in and clean it all up. So there is a brain resident population of mast cells. So we had asked the question, is there a sex difference in mast cells in this preoptic area? Again, we're still in the preoptic area, this region that's so central to male sex behavior. And so Katie Lenz, a postdoc in the lab, just went and counted the mast cells. So this is a low magnification image of the preoptic area. This is high magnification, and this is how you count mast cells. 1, 2, 3, 4, 5, 6, and we're done. And so Katie did an exhaustive analysis of the mast cells in the preoptic area. She started on the day of birth, PN0, went all the way up to adulthood, PN60. This is when they're reproductively active. And as you can see, developmentally, the first 10 days of life, there are a lot of mast cells in the preoptic area, and they are deep in the neural tissue, by the way. They're not just hanging out in the meninges. And there's about twice as many in the male as the female, which gave us a hint that they must be doing something important to have so many there. So she asked the question, are the mast cells talking to the microglia, which, as I said, is what normally goes on in the injury model. Okay, but this is, again, just a healthy brain. And so to do that, we went back to that sensitive period, and she has drugs that could stabilize the mast cells in males, so they stop degranulating, or stimulate them to degranulate in females. And because these cells are so ubiquitous throughout the brain, there's a lot of pharmacology associated with them because they are also the basis of anaphylactic shock. So I always like to say mast cells will both annoy you and they will kill you. So respect the mast cells. And when she just treated them in the first couple of days, and then we looked at we asked, are the microglia listening? Did they change their morphology? And does it change adult behavior? So the top two panel here are the ones I already showed you, with the female on the right and the male on the left. And if you go down to the left-hand quadrant here, this is a male whose mast cells, we just told it, calm down for 2 days, just stop degranulating, so that they couldn't release anything. And you can see they look indistinguishable from the females. And the female here on the right is one who we gave a drug to release the contents of her mast cells for 2 days. So a complete sex reversal, and that's actually quantified here. So what we had done was activated the mast cells in the females and deactivated the mast cells in the males. And then we raised them to adulthood and asked, did this impact their adult sex behavior? And the answer was yes, that it did. I won't go into big detail, but these are normal males and females. Females don't tend to mount other females in rats, much less than in mice. They do more than in rats. But here, if the female had her mast cells degranulated, she was very interested in other females and behaving like a male. And likewise here, this is a normal sex rate. Females don't mount unless they had their mast cells degranulated. But I want to go into all the details, but just to let you know that what the mast cells were doing was releasing histamine. And histamine was talking to the microglia and telling them to make the prostaglandin, and the prostaglandin was talking to the neurons to make the dendritic spines. And so we'd previously shown a role for normal astrocytes. So when we kind of got to the completion of the sex differentiation of the preoptic area, it involved 4 cell types: neurons and astrocytes, which are related, they have common precursors, but also microglia and mast cells, with the mast cells actually running the show. So 2 of the 4 cell types are non-neuronal and are actually bona fide immune cells. Okay, so we've sort of left the sex differentiation of sex behavior behind, more or less, and we're now looking at another system in detail, the medial amygdala. And we were interested in the medial amygdala because of its role in juvenile play behavior. And this is the reason that we've stuck with the rat too, is because mice don't show this behavior. I'll show you what it looks like in a minute. So why play? Well, it occurs during a restricted developmental period, meaning adolescence. It is more physical, intense, and frequent in males, pan species, right? In every species that plays, males are more robust than females, with the exception of the spotted hyena, which has a very unusual situation in which females are dominant anyway. The sex difference is established early by hormones, but when it's expressed, there's no hormones around. So it's really comparing apples to apples. The males and females are in the exact same hormonal status, and it has strong face validity with humans and other species. And of course, we're interested too because it's dysregulated in children with autism, and probably in children who are going to go on to become schizophrenic or other disorders. We don't really understand it very well. This is what it looks like. We put them, they have a play date every afternoon for about 10 days. So it's not their cage mate, it's somebody they just meet in the afternoon in a neutral arena. And that you can see the 2 males on the top, they're having a great time, they're wrestling, chasing each other. The 2 females, they know they're there, they're interested in the other female, they just don't do this kind of exhaustive physical play that the males do. And we have to videotape it so we can go back and quantify it because it's happening so fast. But it has a lot of different components to it. So there's what's called a pin, when one animal pins the other, then there's boxing, there's pouncing, and there's just plain old wrestling. So a lot of complexity here to work with, which is a lot of fun. The neural circuitry of social play, as well as we know, overlaps that of all adult social behaviors. The reason I say as well as we know is because in the 1980s, play was a really, really prominent behavior that behavioral neuroscientists were studying. People like Michael Meany, Bruce McEwen, they had, it was a really, they really appreciated the complexity of it. But then the mouse came along, and with the superior genomics of the mouse, but the mouse just doesn't show this kind of behavior. They're very social, but they just don't do this complex play behavior. What they had discovered in the 1980s is that the medial amygdala was the key node for the sex difference. If you just put testosterone right into the developing amygdala, you would masculinize a female's play behavior. And we wanted to know what the mechanism of that was. About 10 years ago, we published a paper in which we found a sex difference in cell proliferation in the developing medial amygdala. And it was a fun sex difference because for once it was more in females than males, because everything's always more in males, more in males, more in males. And here it was, whoa, it was more in females. And we used BRD labeling to find the number of newborn cells. And the females had about twice as many newborn cells as the male. And then we showed that it had implications for social behavior. We were sure we had done every appropriate control, that this was cell proliferation, and the reviewers believed it too. It was cell proliferation, but we were wrong. It was not cell proliferation at all. What it was is that we had not been paying as close attention to those microglia as we should have, which is a new graduate student came in the lab, Jonathan Van Rissen, and he did the right thing, went to the microscope, just looked at old slides, and he looked at some microglia and he said, "What are those funny cup-like things?" Funny cup, oh my God, those are phagocytic cups. Those are big phagocytic cups. This is some of his earlier imaging. I call this my Baltimore lacrosse microglia. Here on the left, we have a Mickey Mouse microglia. On the right's a double D cup microglia. And our imaging has since improved, but we could see these giant phagocytic cups. So Jonathan asked the simple question, is there a sex difference in microglial phagocytosis just during this sensitive period, that first week of life? And so here is the percent phagocytic microglia. This is a snapshot in time from the day of birth to the end of the first week of life. You can see both males and females have a lot of phagocytic microglia, but much more so in the males. And the level of phagocytosis is actually extraordinarily high, with some of it up to 30%. Again, remembering this is immunohistochemistry. It's a snapshot in time. The whole process takes 20 minutes to 2 hours. So there's just a whole lot of gnashing going on. And then it's clearly developmentally regulated, dropping down to almost nothing after the first week. So of course, the big question is, what are they eating? Well, we were pretty darn sure they were eating other cells. This is not the synaptic pruning nibbling of Beth Stevens, right? these guys are, they're chowing down. So we're pretty sure they were other cells. And in fact, they are. 80% of the cups contain DNA based on DAPI. But the big question was, are they eating the newborn cells? Because we know that there's a lot of cell proliferation going on. To do this, we used proliferating cell nuclear antigen, PCNA, which is a marker of newborn cells. And here we do some MRs 3D rendering, and you can see this microglia has reached out and snatched this brand new cell here in its phagocytic cup. Note also, though, that this is the nucleus of the microglia, which is also PCNA positive, meaning that this cell was just born within the last 48 hours. And this is the best reminder of anything, that this is an immune cell. No cell of the nervous system can differentiate within 48 hours and start being functional, right? So this is a completely different kind of cell in the brain. Here, if you'll let me use a baseball analogy, I don't know anything about cricket, but I call this sort of my stealing the home run, reaching over the wall microglia. We would see this all the time, where they would just sort of shoot an arm out. There's some cell over there they see that they're going to grab and phagocytose. But more importantly is when you look inside the cup at the cell in there, this is its nucleus, and you can see that that nucleus looks quite intact. This does not look like a cell that was beginning the process of apoptosis. 60% of the cups contain newborn cells in both males and females. So they are indeed very preferential towards the newborn cells. So what drives this higher rate of microglial phagocytosis in males? Well, we're neuroendocrinologists. We always do the classic experiment, and we just injected females with testosterone and just asked, is that going to increase the phagocytosis? And indeed it does. That's here on the left. Here's the normal sex difference of female phagocytic microglia, male with more phagocytic microglia. If you treat the female with testosterone, she comes up exactly to the level of the male. Over here are the number of newborn cells. Here's the sex difference. The female has more newborn cells than the male. Treat the female with testosterone and the number of newborn cells comes exactly down. So as you can see, the relationship is completely inverse. And in fact, when we regress them, there is a very strong negative correlation between the number of phagocytic microglia you have and the number of newborn cells that you have. So is this a process called phagoptosis? Was actually coined at your fine institution by Guy Brown and Jonas Naher in this really wonderful review, in which they joined the words apoptosis and phagocytosis to come up with the word phagoptosis. So every cell has on its surface a collection of signaling molecules of either eat me or don't eat me. And most of the time you're sitting there with don't eat me signals. But if you have a bad day, you might say, I really don't think I can go on anymore. I'm in too much distress. And then the cell can actually take enzymes and flip out what are called eat me signals of a variety of sorts. And if you happen to be encountered by a hungry microglia at that time, the cell will be engulfed and actually killed with an oxidative burst from the microglia. So it's a living cell that is going to be engulfed and consumed by the microglia. There's a whole host of these different eat me and don't eat me signals, and they're in a constant state of balance. And there's a variety of different ones to explore. We honed in on the complement system, in part because of what was going on with the synaptic pruning, but for other reasons as well. And that's how the microglia and the cell that is going to be dinner are talked to, is through these transmembrane systems. And so to find out if our cells would have survived if not phagocytosed by the microglia, we needed to stop this process, right? So stop the recognition of the eat me signal by the microglia. And to do that, we could use antibodies that would basically just jam themselves in between the microglia and its target cell and just blanket the eat me complement signal so that the microglia is blind to it. And so we did that by injecting in the first 4 days along with the BRDU, so we could count the number of new cells. And we use the animal as its own control, using a neutral antibody on one side of the amygdala and this phagocytic blocking antibody on the other. And the prediction is that under the neutral antibody, you would have a reduced number of BRDU cells because microglial phagocytosis would go on normally. And on the blocking side, there should be an increase in the number of cells, meaning that these are cells that would have survived if not phagocytosed. And when we did the experiment, that's exactly what we found. Here you can see in the males, this is the neutral antibody side, this is the blocking antibody side, and there are more newborn cells in every single animal with the exception of one. Whereas in the females, it's just kind of higgledy-piggledy, even though it looks like a trend, it's not. We've replicated this several other times. So this is an effect specific in males, and there's reasons why this is that I didn't put in that we can talk about during the question session if anybody's interested. And this is just to show that that antibody is in fact on the phagocytic cups, that it can block the phagocytosis. Well, what were these poor innocent babies going to have become if they had not been phagocytosed? As I mentioned, no cell in the nervous system can differentiate within a couple of days. You need at least 2 weeks to differentiate into the various cell types. And there's all the options are neurons, astrocytes, oligodendrocytes, microglia, or some combination of the above. So we injected with BRDU the first couple of days of life and then waited until they were 26 days old, which is actually the time when they're playing. This is an adolescent. So let's think about like this is like an 8-year-old, and looked at the cell fate. And we did various markers, GFAP for the astrocytes, NUN for the neurons, IBA1 for the microglia. And as you can see, the overwhelming majority, and we should have known in retrospect because it makes perfect sense because there's not a neurogenic niche in the medial amygdala. Overwhelming majority were going to become astrocytes, 83% of them. 13% were going to be neurons, and then 7% microglia. And this actually beautifully adds up to 100%, which was very gratifying. So, and you can see also that the females wind up with more astrocytes than the males in the medial amygdala. We're just looking in the medial amygdala, right? Just this little node. And then in fact, we went back and confirmed that the male microglia were preferentially eating precursors to astrocytes using a marker called ADL-H1L1, which is, these are cells that would have become astrocytes. So are those astrocytes future regulators of adolescent playfulness? Is this the key to why males play more? And to do that, we went back and repeated the antibody experiment, only now we put it on both sides of the brain. And first we confirmed that it does affect the number of astrocytes. Here's the sex difference with females, more astrocytes than males. Unless we blocked phagocytosis in the males, they have more astrocytes. And then over here is if we allow them to come up to age to play. Here's the sex different play, females low playing, males high playing, except for the males who have now a higher astrocyte population because we blocked phagocytosis. And we're doing experiments now in which we're looking at the activity of the GABAergic neuronal population, and the astrocytes we believe are regulating the neuronal activity. It's not a direct effect of astrocytes, but they're gating the activity of the neurons. We still haven't explained why microglia are more phagocytic in males, and this is a question that we're still asking, but to sort of get at that. It's, as I sort of said at the beginning, we used to think microglia just sat around. We called them quiescent, and that they were just waiting for an accident to happen. But when we came along with all these amazing imaging and the genomics of the mouse, we discovered that actually they're really busy. They're moving all the time. And that I usually show an image of the mouse here, but we actually recently developed a CRISPR modified rat in which the microglia are expressing GFP. So these are actually microglia and a slice of rat brain. And as you can see, they are always moving and they're always checking everything around them. So we created an interactome in which we looked at how we measured the tip of the microglia to the most adjacent cell. And this is just a heat map of distance from the tip of the microglia, and asked at a snapshot in time, is there a sex difference in the number of cells that are being contacted between males and females? And as you can see here, measuring lots and lots of cells, males on average are reaching out and touching more newborn cells than more cells than females. But when we look at the newborn cells, we find that the males are actually touching more newborn cells. Data I didn't show you, these newborn cells are coded in complement, coded in EE signals. So the question is, are the males just encountering more newborn cells stochastically and thereby engulfing them? In other words, maybe the male microglia are just more fidgety than the female microglia, which would fit perfectly, wouldn't it? So I'm just going to then real briefly return back to the preoptic area for one little more just so story to just kind of really bring home this variety of ways that the immune system can affect the brain. So as I said, this is the key brain region for male sex behavior. It's also the key brain region for another aspect of mating behavior, which is partner preference. And it's the site of the oldest, most well-characterized sex difference in the brain, a sexually dimorphic nucleus, named for the fact that it is 3 to 4 times larger in males. It's actually visible with the naked eye once you know what you're looking for. It has a human analog named ENA4 that was made famous by Simon LeVay back in the 80s when they found it was correlated with male homosexuality. So its volume is correlated with partner preference in humans, mice, sheep, and rats. It's been really hard, though, to directly functionally tie it to partner preference, but it is the well established that this sex difference occurs because more neurons, or because the same number of neurons are born in males and females, and then they die in females because they're not getting that growth benefits from testosterone and aromatized estradiol. And basically the female neurons just die of starvation, right? That there's some secret sauce, some growth factor that they're not getting. And that's been established since the 1970s. It's never been questioned. And I am here to tell you that it is wrong. And again, it's because of the microglia. So Lindsay Pickett in the lab said, well, I'm going to go back and look at the microglia just in the vicinity of the sexually dimorphic nucleus. This is the whole medial preoptic area. This is where the sexually dimorphic nucleus is. And she quantified the phagocytic microglia across the first whole almost 2 weeks of life. The sex difference in the SDN doesn't occur until about PN8, at which time she saw there was a much higher level of phagocytosis in the female SDN than the male SDN. So again, the question was, what are they eating? Well, in this case, it was not astrocytes. In this case, NuN is a marker of neurons. They are in fact eating neurons, and up to 80% of the cups contained evidence of being neurons, and it looks like they taste really good too. In this case, this is after the oxidative burst and they've killed the cells. So she did the same experiment where she was going to block the phagocytosis and ask if that would functionally increase the size of the SDN because those neurons weren't killed off. And so she did the blocking antibody 3 days prior. And this time she did it on both sides of the brain. And in fact, she could increase the size of the SDN in both sexes. So here's the normal sex difference, male to female, 3 to 4 times larger. And if you just stop the phagocytosis, stop the microglia from munching, the neurons survive perfectly well, and you have a bigger SDN in both the male and the female comes up to the male range. So the real question is, does that functionally impact partner preference? And we used an odor preference, a 3-chamber olfactory preference test for a male versus a female versus a neutral. And just to, go quickly, this is the normal. Females really prefer male odor. Males don't prefer male odor. This is time spent with the male bedding. But the females that we blocked the phagocytosis and increased their SDN lost their preference for the male bedding. We don't know what's going on with these males over here, why they seem to have shown a increase in preference for male bedding. So these are ongoing experiments that we're still working out, but this shows kind of our a toolbox of things that we can use. So in summary, I showed you 3 cases of immune cells basically running the show. Mast cells can activate microglia to make prostaglandins and induce synapses in the male medial preoptic area to masculinize mating behavior. Microglia can engulf astrocyte precursors in the developing male amygdala to masculinize playfulness, and microglia phagocytose neurons in the sexually dimorphic nucleus to regulate partner preference. So all of this is in healthy brains. So what about in a disordered or diseased brain? As you all know, being male is your leading biological predictor of your relative risk for a developmental neuropsychiatric or neurological disorder, but also early life inflammation is the leading environmental predictor of your relative risk of a developmental neuropsychiatric disorder. So are these two facts related? And this is just a review paper by Donna Whirling based on her reanalysis of a transcriptome of fetal brain and adult male autistics and typically developing autistics. And she found that there was an overlap in sex differential regulation of genes and autism spectrum disorder risk genes. And they seem to hone down in on microglia and or astrocytes. And if you read the original paper, it's almost like you can see she was puzzling as to why were all of these immune cell genes showing up in her male specimens. And then she went to the animal literature, and then she found in the animal literature, my work and others, showing that, yeah, this actually makes a lot of sense. It wasn't just a spurious that she happened to have sick male fetuses kind of thing. And then she confirmed that it was also true in the adult autistics, that there was more inflammation activated genes. Why this is the case is another question. We can maybe talk about that during the answer period. So finally, my conclusion and speculation is that factors are normally associated with inflammation or injury are elevated in multiple parts of the normal developing male brain, making it vulnerable to early life insult. And in work that I didn't have time to talk about, we think that epigenetic suppression of inflammatory genes may provide protection to females, so that it's sort of a two-edged sword. And thanks to the funding agencies. This is my lab 2 years ago at Halloween, where we all dressed up as our favorite immune cell. You can see this is Lindsay Pickett, a mast cell over here. She kept running around degranulating. I'm a stem cell. These are the microglia people back here. Here's a natural killer cell, and so this is a B cell. So anyway, we had great fun actually pretending like we're immunologists. And these are their actual names. Everybody in red contributed some data to this. So thank you very much for your attention. I hope that we still have time for questions and answers.